scientific paper, the structure and folding of acidic nuclear protein pp32 is investigated. pp32 is a protein commonly expressed in tissues with the function of being a tumor suppressor that inhibits oncogenes. Oncogenes are responsible for promoting cell replication. Over time, they can become mutated, and if not suppressed by proteins such as pp32, cancer can occur. In looking at the structure of pp32, this protein is a repeat protein known to have several portions of its gene sequence that code for tandemly repeated structural motifs that contribute to its secondary structure, such as alpha helices, beta sheets, beta turns, and beta barrels, that then come together to form a folded domain. This makes up the final shape of the protein, known as its tertiary structure. Proteins then maintain this folded nature through covalent interactions as well as strong non-covalent interactions such as hydrogen bonding, van der Waals, and ionic bonding. For pp32, the most common motif is a beta strand that contains several leucine-rich regions. In looking at the structure of leucine, leucine is a hydrophobic amino acid in which it does not like to interact with water. This hydrophobic nature is due to the side group that is composed solely of carbons with no polar molecules. These leucine-rich regions cause the beta strand motifs to fold in a way that the leucine residues are folded inwards to shield the hydrophobic portions of the protein away from the aqueous environment. This follows the hydrophobic effect of protein folding. To also aid in protecting the hydrophobic portions of the protein, as well as aid in structural and folding, the leucine-rich region proteins have N-terminal and C-terminal caps. The terminal caps of the pp32 protein have very similar sequences. However, the portion of the pp32 that codes for the C-terminal cap has 5 additional amino acid residues that the N-terminal cap lacks. This can be seen here in Figure 1. The C-terminal cap shows the sequence pattern where X represents any residue and phi represents the hydrophobic residues. These bold residues of tyrosine and aspartate represent the residue of the sequence that interact with each other, forming a hydrogen bond. The C-terminal uses these 5 additional residues to create a beta hairpin structure, which varies from the N-terminal cap that lacks this beta hairpin. To test the effects these caps had on folding and structural stability of pp32, several tests were performed, implicating several methods to study unfolding and stability.
In the methods section of this paper, 5 different variants of pp32 were used. 5 residues were removed from the C-terminus. Tyrosine 131 was removed as the H-bond donor. Glutamate 146 was removed as an H-bond acceptor. Both glutamate 146 and tyrosine 131 were removed, and also the N-terminus were removed. Using 3 different stability and unfolding methods. One was urea-induced protein unfolding. This was used on all the different variants to unfold the proteins. And urea works by forming hydrogen bonds with the exposed amide groups of the protein backbone, which are stronger bonds than the protein's non-covalent interactions. To quantify the unfolding, the free energy change, delta G, were used to express the protein stability. The free energy changes were measured under the assumption that less negative values equal less stable proteins. NMR spectroscopy was the third tool used to show protein structure. The same pp32 variants were examined using NMR to measure their structural changes. The normal pp32 shows a peak at 217 nanometers. If any peaks were to show up above or below this number, then it can be assumed that structural changes occurred.
The results of this experiment testing whether the 5 additional residues of the C-terminal cap sequence had affected the stability and structure of the pp32 protein used variants to compare secondary structure, stability, and folding properties. In using the far UV spectra for both the wild-type pp32 protein and a variant lacking the 5 additional residues, as Figure 2a shows here, both show a minimum curve at 217 nanometers, which is characteristic of beta sheet structures in proteins. When comparing the curves, the wild-type pp32 shows a stronger negative curve that indicates the beta hairpin is occurring. In comparing a variant containing only 145 residues of the 154, the curve drifted closer to 200 nanometers, which indicates more of an unfolded protein. From this, it can be resulted that residues 146 through 149 are needed for the protein to maintain its folded structure, while 150 through 154 are not. To look at the effect of the 5 extra residues on stability, the protein was induced to urea unfolding, which was monitored by far UV CD. Looking at figure 2b, we can see that the curves are sigmoidal, indicating that the free energy was dependent upon the urea concentration. Looking at the curves, we can see pp32 wild type needed a higher concentration of urea in order to be unfolded, indicating this protein was more stable than the variant with only 149 residues. In looking at table 1, which shows the numerical data for this experiment, the M values are almost identical, suggesting that these proteins are both unfolding similarly and are about the same size. However, when looking at the free energy changes, the wild-type pp32 has significantly higher values, indicating that the 5 C-terminal residues do contribute greatly to the stability of this domain. To determine the effect the hydrogen bond had on pp32 stability unfolding, the amino acid residues that contributed to this bond were replaced with other residues that could not form a hydrogen bond. When comparing the structures through far UVCD, it was observed here in figure 3 that the peak at 217 occurred, indicating that the removal of the bond did not lead to protein unfolding. To test the stability, pp32 without the hydrogen bond, the variant was induced to urea unfolding that was monitored by far UVCD. In figure 3D, we can see that the curves indicate that the wild type is the most stable as the curve is shifted to the most right. In table 1, in looking at the free energy changes, all the values had decreased, becoming more positive, indicating that the amino acid residue substitutions were all destabilizing. This indicates that pp32 is stabilized by the capping hydrogen bond, as seen by the most negative free energy change.
To understand the role of the N-terminal cap, 18 residues prior to the first repeat were removed and compared to the wild-type pp32. The results showed that the N-cap removal of the pp32 has similar shapes to the pp32 wild-type, but the N-cap removal of the pp32 had much lower delta G values compared to the wild-type pp32, showing that the N-cap removal destabilized the protein. So, in conclusion, we see in this article that deletion of the C-terminal equal total misfolding of the pp32, whereas deletion of the N-terminal equals some changes but overall conservation of the shape for the pp32 protein. This showed that the pp32 begins its folding at the C-terminus. This is opposite to most proteins, therefore it is hypothesized that a molten globule must be formed to assist in the protein's folding. However, both terminals are essential to the formation of this molten globule. The C-terminus is very important to the molten globule formation. The pp32 protein serves a vital function in the human body as a regulator of transcription, apoptosis, phosphorylation, and cell cycle progression. The pp30 protein is heavily involved in hyperphosphorylation of the retinoblastoma oncogene that directly inhibits the cell cycle. The pp32 protein inactivates Rb by hyperphosphorylation, which induces the cell to commence the cell cycle and block apoptotic signals. This mechanism has been shown to be evident in many cases of breast cancer and prostate cancer, and pp32 is believed to contribute to cancerous growth in mutated individuals. Bridging the gap between the gapping motives and the human cell functioning, the pp32 folding structure determines its function. If one of the terminals of the pp32 protein were to be misfolded, then the pp32 could have a gain-of-function mutation. The article mentions that the pp32 protein is prone to mutations, which could cause the pp32 to be a major contributor to many cases underlying cancer in humans. The area of interest would be the mutations not to the C-terms, because this would cause complete misfolding, but to the N-terminus because it maintains a secondary structure but could have increased activity of the RB oncogene. Thank you for watching.